And now we're going to be talking about the challenges associated with manufacturing gene-modified cells. So in these products, you're introducing your gene therapies within cells. So the genes are being delivered in their own little protective bubble, as you will, and there's challenges around that. First, we can talk about some of the advantages. I thought I would do that, of introducing genes in cells. First, you have enhanced delivery of payload, not just enhanced delivery, but control of delivery. You can assess the efficiency of the transduction or the transfection of the cells before you administer them, and you can control that. Also, you can anticipate quicker action of the product, perhaps, because the cells come into the body locked and loaded, as opposed to delivering a virus that has to first find the cells and get into the cells and then make the gene of interest. And so right now we're going to move into talking about some of the challenges of making these products. We have our panel here. I'm going to give them each a minute to introduce themselves, and then we'll move into some of the key areas of manufacturing these products that are on our minds. We talked about this last week, some of the themes in this space, and we think are probably on your minds as well. Let me start by, sorry I didn't introduce myself, Jessica Carmen. I'm the Director of Business Development at MaxCyte. MaxCyte has a non-viral platform for the delivery of genes into cells, and we also have our own therapeutic programs. We'll be talking about our company and our products later today, so I'm not going to belabor that. But first, let's start with Aldevron.
My name is Al Dietz. I'm director of the Impact Lab. That stands for Immune Progenitor and Cell Therapy Lab at Mayo Clinic, Rochester. The lab is a GMP facility that has primarily responsible for managing any Mayo Clinic investigator-initiated INDs, as well as developing our own platform technologies, and we've done contract manufacturing. I think we're approaching our 300th patient, autologous patient under IND, and about over 1,200 products we've infused into patients over the last 19 years. We haven't done much in the CAR T-cell space, but we have done modified measles virus and some RNA transfected approaches towards altering cells.
And I'm Stefanos Techaris. I'm a Senior VP for Corporate Development at Cell Medica. We are a T-cell immunotherapy company. So we're a T-cell immunotherapy company based in London. We have facilities in Houston and Zurich as well. We've traditionally been working with T-cells against viral infection, including oncology. We have an EBV product for EBV-positive lymphomas. We've more recently ventured into the world of gene-modified T-cells. We have a very exciting platform as a collaboration with Baylor College of Medicine that's based on a subset of T-cells, which is invariant. They're called the natural killer T-cells. And we're building CAR products based on these cells. One great advantage of these cells is because they're invariant. They don't target human antigens. They therefore don't cause graft versus host disease. So we can therefore develop off-the-shelf allogeneic T-cell, CAR T-cell therapeutics without the need for gene editing. We don't need to remove the TCR out of these cells. So obviously simplifies life a lot and makes a product much more consistent and much easier to manufacture for off-the-shelf application. Our second platform is, again, another collaboration with University College London, just down the road from where we're based in London. And that's based on TCR-modified T-cells. It's the dominant TCR platform that was developed by Professor Hans Staus. It increases the expression and the function of TCRs being expressed in T cells, and therefore leads to much higher levels of antigen-specific activation at lower levels of antigen presentation, which is common in tumors. So we're also very excited about this platform. We consider it to be one of the, hopefully, one that will become an important component of TCR modified T cell therapeutics. I'm here to discuss, how we put these genes, these CARs, and these TCRs into these cells. As a new entrant in the field, there's a lot for us to learn. Initially, one could easily say, well, you just need a vector to express the genes, don't you? But it's proven, of course, as we knew, that it takes a lot more work. And a lot of that work is going to happen through partnerships with experts in the fields. There are a lot of very good companies out there that specialize on different types of gene delivery, and we're certainly interested to learn more and find a suitable partner that will help us optimize this aspect of the manufacturing.
So I'm Natalie Mount. I'm the Chief Scientific Officer at Gamma Delta Therapeutics. The Gamma Delta Therapeutics is a new spin-out company. We're formed formally actually in February of this year, when myself and the rest of the management team joined the company. And Gamma Delta Therapeutics is really about exploiting the potential of gamma delta T cells, and particularly tissue-resident gamma delta T cells, as a new approach for immune therapy, and particularly in immune oncology. Compared to alpha beta T cells, gamma delta T cells have advantages, so we can really combine the power of what we learned through alpha beta T cells, but put them now in a platform which is not MHC restricted, similar to what Stefanos was saying with his NKT cells. Gamma delta T cells are also not MHC restricted, and therefore can be used as an allogeneic platform in order to deliver cell therapies. Tissue-based gamma delta T cells are also very well matched for being living in a hypoxic environment. They like to live deep in the tissue, so the approach really is very well suited for solid tumours, which is one of our first targets for our immune oncology platform. So the company is a new company. If people want to hear more about it, I'll be talking about it tomorrow during the main agenda in the company presentations. But we're a preclinical stage, did a major deal with Takeda Pharmaceuticals earlier this year, and we're now moving towards the clinic with a platform of cell-based medicines based on the gamma delta T cells.
All right, so a couple of topics that are relevant for the very complex manufacturing process of gene-modified cells. When we were talking, we talked about, we had a conversation about the impact of early decision-making in your manufacturing process as these technologies are sort of coming together and people are bringing ideas to reality. Decisions are made, and there are long-term impacts of the decisions that are made early that oftentimes developers don't realize. And so you have some unique
yeah, thanks. So we have kind of our philosophical approach towards how we develop our programs. We have developed a number in-house. We've also consulted on some, and we've also worked with companies who are doing, we've worked as a CMO. Typically, we try to approach it, and there's 4 stages that we kind of have broken apart. First is discovery. We're not usually involved with that. Then optimization, validation, and finally manufacturing. If I had to walk home to Minnesota and the distance was what you have to put into effort, I would walk from here to Nebraska or Iowa on the optimization step. Okay, because by the time you get the optimization step, you need to actually have everything fairly ready to go between what you need to do from the concept of discovery to where you're trying to manufacture. The unique aspect of cell viral combinations are the fact that actually you have 4 variables that you have to keep in mind during all of your optimization aspect. One is the vector, or however you're going to do the vector, and we heard some really good comments earlier. It's often not the vector itself that's the problem, it's the process around the vector that's the problem. The cell themselves, which is also something that has to be uniquely optimized. The 2 that are often overlooked are the patient and the patient population. It's amazing how many people will bring a discovery for a new disease in which they've never actually tested that discovery in the new disease in any way, shape, or form. So, for example, when you isolate granulocytes by do a mononuclear cell prep in a cancer patient, you'll often have degranulated granulocytes in that prep. Things like that, which are unique to whatever patient population you're looking at. And the last one is logistics, considering what the logistic aspects of your delivering of your manufactured product are. So you have a variable, you have 4 unique variables, each which interplay on themselves, that have to be optimized prior to actually moving forward. We try to go through the unique steps we try to go through the steps reviewing the GMP-able nature of the availability of the specific products and the resiliency of the process that underlies it. So we do a tremendous amount we try to do as much as we possibly can on the optimization aspects of the process itself prior to moving the product forward. If you don't, what you end up doing, and it's human nature, everybody who has their discovery, every company that has just got their money, everybody that wants to get moving wants that first phase 1 trial data now. But I can tell you a tremendous amount of money is wasted after that first patient has been enrolled by re-reviewing and re-optimizing steps that never should have been done before. The other thing you have to keep in mind is generally we're talking about autologous therapies. So we don't actually make in a 20-patient trial, we don't make one product, we make 20 products. We got to make those 20 products exactly the same, or we introduce noise into the trial that's already too small and usually underpowered anyway. So we got to be we can't find ourselves into that manufacturing thing with a question on whether or not we're going to meet that the certificate of analysis. Or while we met the certificate of analysis, the technician has seen something they've never seen before under the microscope. That's a disaster to me. Okay, so we try to avoid that disaster by putting as much time as we can on the optimization steps.
That was a lot to digest. Really,
we only have a little bit of time.
Really great summary. Actually, I think maybe if there are any questions, because I think there was kind of a lot there. Does anybody have any questions now or any comments about what we just talked about? The 4 aspects of optimizing early? No? Okay. Everybody's... Is there a question?
You have a question?
No? Okay. All right. Keep digesting. Keep thinking. Stefanos, I wanted to talk about the potential for long-term patient follow-up. I thought it was very relevant to mention that considerations around the patient and how each patient is unique and therefore each batch is unique can really complicate the development of these products. And when we were talking, we talked about the potential for long-term follow-up of circulating modified cells in patients. And as we all know, Novartis is going to be paving the way with these products being the first product to have been approved. And we all can only guess at what it's going to take to follow up in these patients. Do you have any thoughts about that?
Well, certainly the Novartis ODAC meeting about a month and a half ago was very illuminating for all of us. And obviously we all need to learn from the ones who are paving the way, the front runners. Of course, we can't always assume that they get everything right, but there's a lot of trial and error in optimizing these. There's regulatory aspects to it as well as the efficacy, and certainly with CAR Ts, persistence has been linked to efficacy, rightly or wrongly. The CD19 T cells tend to persist longer in patients that tend to respond, and there's potentially a correlation there. So being able to at least understand whether there is a correlation or not, and try to understand what are the components of a successful, at least TCR or CAR therapeutic, is important for us. So from that perspective, we need to generate as much data as we can out of the patients in the early trials, and all of that information is going to link back to then designing the next trial or seeing whether there's any optimization in the actual product. Likewise, things like transduction efficacy, how important is it? How many transduced cells does one need for efficacy to occur? Again, that's one that's not clear. I didn't see a strong correlation, at least with the Novartis data. And I'll keep coming back to Novartis because they've obviously shown a lot of that data. They've shared a lot of that in that meeting, and they've treated a lot of patients, so they should know. And then obviously there's regulatory issues and safety issues. So being able to monitor the patients for long term for the persistence of these cells, as well as the potential for the replication competent virus, retro or lentivirus, is an important consideration. And it's one that we probably are going to have to do for a while. There may be a world in the future where we actually have to screen the product for RCR or RCL for every batch. And potentially keep following the patients until we know a lot more about the safety implications there. So the way I'm seeing it is that we keep moving forward and we keep breaking new ground and we keep finding out more important parameters that we need to keep in mind. And that goes to some extent to the concept of optimizing early, which I think has to be approached with some caution. I wouldn't want to claim that prior to starting my phase 1, I have finished the process, and that's the process. We all know that there will be improvements, and we will learn through performing these trials areas that we need to improve. But also, in order for us to be able to treat thousands or hundreds of thousands of patients. We all have to move towards an automated world where things happen in a more systematic way. And before we get to that, obviously you implement a suboptimal process, you'll end up with an suboptimal automated process, which isn't going to help anyone. So the automation, the improvements, the optimization all have to keep happening. And I'm afraid it's just going to have to keep happening even while we're performing the trials. And either informing the actual product that is being optimized, or obviously follow-on products or products against different targets that have additional optimizations included in them. So it's going to have to be a learning by doing to some extent, but obviously trying to optimize everything that we can early on. We absolutely should.
Absolutely. Again, I'll open it up to the panel. Or the audience, I should say. Does anybody have anything to comment or add onto that? This is a workshop. We're all meant to help each other
and
share all of our experiences and our concerns about the field, not just us up here. No? Okay, keep drinking coffee. It's going to come. So Natalie, when we were talking, you mentioned that you have experience on assessing comparability through early to late. I think this is a very important concept that we need to talk about because, as Stefanos mentioned, the process is going to evolve, and we would be foolish to think that we have our process locked down in phase 1 and that it will not change, because if nothing else, the technology is changing around us. The tools providers are getting more savvy. The regulatory agencies are getting more savvy as more products are coming through. And so even if we think our process is as good as it should be, the world around us will change and we will need to change with it. So if you could talk about your thoughts on comparability.
Yeah, sure. So, comparability is, it's a big deal for us in cell and gene therapies, and for the reasons we were just talking about. So, what is comparability? So, comparability really is the requirement to show that when you make a change to your manufacturing process, whether that's to your drug substance, the drug product, manufacturing site, anything about how you're manufacturing or where you're manufacturing the product, but actually that doesn't have an adverse impact on the quality, safety, efficacy of the product. It doesn't mean the product has to be identical to how it was in the previous process, but it's important to have data that actually are predictive of what changes that are going to come about and, demonstrate you don't have an adverse impact on quality, safety, efficacy. So comparability is a big deal to us in cell and gene therapies because, as Jessica and Stefanos were just talking about, we know we're going to have changes. We know when we start from an initial process, that we're going to be suboptimal in lots of ways. We may not be able, we'll need to change scale. We probably need to change technologies, the bioreactors. We may learn from the early studies and need to change actually components of our product, or know we need to tighten specifications around particular components. And we also want to change things like cost of goods and general scalability and improve logistics. So there are lots of things that we'll need to change as we go through. And it's not the case that we can expect that we can lock our process at a particular point and never change it again. It's going to be a changing process. It's kind of inevitable in a field that's moving this fast scientifically and also moving out to larger patient numbers. So comparability is really important. So for cell-based therapies, really the successful approach to comparability is really anchored in cell characterization and actually understanding the product. Understanding your identity, purity, potency, and working out from the early stages what are the critical quality attributes of your product, which then need to be pursued all the way through any changes you make to the manufacturing process. And obviously that's going to be it's going to be a learning curve. And I think really important in early development is actually to characterize the product as much as you can and really to have a good scientific understanding of the product. There's an opportunity in the early phases to really learn about the product. You don't have to put all the everything you know into formal release testing, but you can add things for information, and as you're developing the product, you can explore the components. And that will give you a good idea, actually, building towards what are your CQAs, but actually also a broader understanding of the product. An opportunity to use some of the more, scientific-rich approaches. You might want to do mRNA, you might want to do mass cytometry, you want to do a whole secretome analysis, so you really understand what's going on with the product, and from that then you can start to look to funnel down to what are the key attributes that you want to follow. And obviously then as you go through development, you're going to need to focus down on a very small number of key attributes that you're going to follow and actually formally release against. And the approach to characterization also does depend on whether it's an autologous product or an allogeneic product. In an autologous product, as we know, there's inherent patient variability. So what we're trying to do is understand what's important and also make sure we understand how a manufacturing process, what component of variability comes from the manufacturing process, and that's what we want to minimize, but on the background of what's going to be inherent variable patient samples. With an allogeneic process, obviously that variability is inherently less at the start. It's still a natural product, so it's still going to have variability, but we can look to minimize that. But importantly for allogeneic products, we need to be thinking about having a banking strategy, master cell banks, how many we're going to use, and how are you going to demonstrate that when you need to shift from one master cell bank to another in the future, actually, you're able to show the comparability between those two products. So I think investment in analytical assays is really, really important to enable cell characterization, which then really enables the future strategy. And with analytics, with our immune cells, for example, we often do a lot of assays like flow cytometry. So again, it's looking at actually how can we kind of standardize and try to cut out some of the variability that will come from operator analysis, for example, trying to get towards defined protocols for analysis of something like flow cytometry, trying to move away from visual observations of cells to actually something more quantitative, so you can get the parameters defined down. So that there is a regulatory framework around comparability and what's expected. A lot of the things for cell-based products are actually within ICH Q5E, the comparability guidance for biologics. So a lot of those things directly translate across. The need for a risk assessment, actually defining what you're going to change, doing a risk assessment, doing a formal comparability study, then looking at analyzing that, and then look at the impact on the CQAs. So a lot of it fits into that basic framework. So I think moving across regions, you can expect within the ICH regions that there's a consistency to how people will look at comparability and what's needed. But I think it's also important to understand that right from the start there are differences in regions between actually what they require in terms of total analytics. So, for example, starting materials are what's required there, how they're regulated is quite different in the US to the EU. You've got different requirements around how you might do donor testing, how you might do traceability. So those kind of requirements, particularly if you've got an allo product, need to be thought about really early, because you don't want to build something into your master cell bank, which means that later on you can't actually translate to a different region. So I think, as Stefano said, regulators are becoming much more savvy about this. They understand, they're seeing products move around. There is a framework which we can work to, but fundamentally, I guess comparability comes down to a really good understanding of your product and really making use of the early stages of development to get a really good scientific characterization of that, and that gives you a platform then of information you can build on as you move forward.
That's perfect. If you don't understand what your product is or what it does, then how can you understand how changes you make impact it? That's a very good point. Are there any comments? Has to be thrown.
Okay, wonderful microphone. So I don't think anybody would disagree that sort of comparability comes down to ensuring consistency within your critical to quality attributes. I guess the big question is, how do you define what is a critical to quality attribute? Yeah. And especially in the era of omics. Mm-hmm. Because you mentioned, mass cytometry, transcriptomics, sequencing, mm-hmm. etc. There's the potential to get a lot of information that is effectively noise, mm-hmm. doesn't matter. So what guidance could you give us with regard to dissecting what's really critical from what's noise? And I know that's incredibly difficult to generalize, but just if you can. Yeah.
I completely agree. There's a lot of, when you're using the early stages to really try and characterize the product, you can generate a lot of information, and a lot of it will be noise. I think I agree with that. I think the question is, as you go through those, you will start to see things actually which you recognize to be important. So the idea, building the CQAs does come from that understanding, and you may understand from the start that the identity, usually you know what the cell product is you're trying to make, but you also know there's going to be variability in that in terms of the percentage of that particular product. Some people's product may be a very much uniform single cell product, as other people's product actually is not. So in T cells, for example, we have a mixture, maybe alpha beta cells, gamma delta cells, NK cells. Then if you go into alpha beta cells, we've got central memory cells, naive cells, effector cells. That actually the products are quite heterogeneous. So it's really trying to understand, from early on, actually, what are the characteristics of your product, and then as you put those through your in vitro assays, when you're looking at potency, for example, you can start to see are there correlations between, my product has a high expression of a particular checkpoint inhibitor. Does that make a difference actually to how it performs in a cytotoxicity assay? Does that make a difference to how it performs in an in vivo model? And in the end, when you get to the clinical data, if you're collecting this information on parameters you think might be important, then you can start to look at correlations. So yeah, I think I agree with you, you'll generate a lot of information and you're not going to want to collect everything, and you can't. But at the start, I think when you're starting out, you can collect a lot of things and look through them and use your knowledge of the product and how you expect it to work, and try and build that understanding, and then you test those things out in the in vitro and in vivo assays as you go forward, and try then to narrow down to, okay, these are things I think are noise I'm not going to worry about, this is the thing actually I think can be important and I'm going to monitor.
And I think you have to, I talked about optimization, and I was talking exclusively about optimization of the product. And I really would stay fairly firm that the optimization stage should stop as much as possible during your first set of trials, your first trial period. But, however, there's platform optimization. And platform optimization can actually, what you should do, at least what we try to do, is exploit everything we possibly can on that first trial to learn what we can about our own process, learn what we can about the patient, and learn what we can about the product. Okay, so I couldn't emphasize more what I'm hearing, which is we really don't, the truth is we don't know what our product is most of the time. And I can say that because I'm not answering to investors. So I don't know what I'm doing a lot of the time. But I can sure try to use what I get to figure it out. And it doesn't mean we're not justified trying this in a clinical trial. We're usually addressing really unmet clinical needs. So I'm fully comfortably, morally and ethically being able to justify the use of these products in patients. They need them. We think it works. We think we can do it safely. But we don't know everything about them. And we should do what we can to learn everything about them. So if I was moving investment chips around, one of the big set of investment chips I would be into is trying to understand the products as top to bottom as much as possible, the variability of the patients that are coming in. So while we can get down to 16 different, we have 10 color flow. We can get a 10 color flow characterization on whatever cell we're trying to manipulate. That's still not a 100% uniform population, and that population is still affected by whatever patients we're getting them from. We need to really understand what we're using as our starting product, what the product is, and what environment it's going back into. And that is platform optimization. And I think strategically, from a company point of view, if I was in a company, I think you have to look at both things separately. What is the optimization of your first process you're doing, and then overall, what's your optimization of your platform within your company and where you're going to go with it?
And yeah, I completely agree that the difference between optimizing and understanding the process versus the platform optimization that needs to continue happening. I just wanted to mention the potency aspect of it, and that's one that we talk a lot about in this field for the last decade or more. And again, what is potency? How long is a piece of string? I think this is something that we're working on, obviously, and we're developing potency assays, and we're trying to simplify and then in the future automate potency assays, because eventually this would be expected to become part of the specifications of the process, of the product. Sorry, but then at the same time. And we're quite lucky at Cellmedica. All of the work that we do is in partnerships with academics, most of whom have been working for 20 or 30 years in the space. So they understand, at least in the T cell space, they understand the immunology. They understand how T cell populations, which are a collection of very different cells in different stages of development and activation. And that's really important in having the potency on the one side, which will have to then be correlated with efficacy in the clinic, and also the science, which tells you that T cells that express these markers or don't express these markers are more likely or less likely to be functioning. And the knowledge has to be evolving in parallel. And of course, then there's the general characterization where you try panels of genomics and proteomics and everything else that you can come up with that characterizes the product. And so with these three aspects, so the one is the end result, the potency, the efficacy, then it's the science, what you understand, what you expect would be the case, and then the sort of broader fishing expedition, trying to find anything else that correlates. All of these grow in parallel, and every time we manufacture a new product from a different patient, we learn something more. Every time we treat the patient, we learn something more. And so these three pillars, I could call them, are going to just become stronger and stronger and enable our understanding to keep developing better products. So the next products that we're going to be developing maybe in 3 to 5 years after the first wave has come into the clinic is going to be a lot more informed based on that information. And that will just continue. And at the same time, the platforms, the manufacturing technologies, the automation of the assays is going to keep growing in parallel as well. So eventually we end up with something that's considerably stronger than what we have now.
Yeah. Potency is a great question. One of the ways we do a lot of work with autologous MSCs, adipose-derived MSCs, and from hundreds of patients. And so one of the questions is, how do we try to identify what markers can be used? And I think what we're trying to go at for a philosophical approach is to what they are not. So we're doing a lot of RNA-seq, and we're doing a bunch of work where we're classifying series of other cell types and saying, well, we know they're not. It's a little like being in an art gallery. If you're looking at only one picture and going, what is it? It's much easier to describe it when you look around and go, okay, it's not that. So we're trying to list what are the characteristics that differentiate it from the cell types that we don't want, and trying to get to our potency assays by more of a process of elimination than try to really do the correlations between what it is and whether or not it works.
So it's interesting I think potency is obviously could spend a whole day on because it's so complicated. And I wonder, specifically talking about gene-modified cells, is there actually a gimme there? I mean, is there one potency that's just, evaluating whether or not the modification exists in the product? Is that just a gimme? Okay, everybody's got to evaluate that. Or is that even necessary? I mean, are there people who are developing, or companies, or entities, I should say, who are developing gene-modified cells who don't worry about assessing whether or not the modification they're introducing is there? And what do we think the regulators will think about that?
Yeah.
It's easy.
If it's just the gene, it's easy. When you put a gene in a cell, then it becomes complicated because you're adding two variables together to get yet a third, which means you not only have one, you have the other one, which by definition is going to come from a patient. And even though we can isolate it and be fairly confident in its purity, the combination of the gene into the cell is going to be a brand new third product. And so it's not as much of a gimme as it sounds. It's better than some things, but if you can, and it's hard to do very general response to that, other than to say that you're going to be combining to get a third product, and that third product's going to have to perform. If you depend on that performance to only be demonstrated within the patient, now you've got to find correlative potency assays in vivo that are going to reflect what's going to happen in the patient. So I wish it was a gimme, but I haven't seen any gimmies in this business yet.
I'd say the transduction itself is more of a phenotypic rather than a functional aspect of the product. Yeah. It's easy in T cells. T cells kill target cells, so that's an easy potency assay. Do they kill your tumor cells? And that's something that at least in immunology people have been performing for many years. And assessing how that happens isn't really rocket science. Of course, that's not the end of your potency. You still don't know. How long do the cells survive? Do they migrate into solid tumors? So can they extravasate and infiltrate the tumor tissue? Do they kill the tumor tissue while they're there, or do they get immunosuppressed? As a lot of things do when they get into the tumor. So this is just something that you can test before you infuse the product, but we still need to understand all these other hurdles that the product has to overcome in order to achieve efficacy in the tumor. But at least from that perspective, potency is important. Then the functional aspects of it, does potency correlate to transduction efficacy? Yes, no. Is transduction efficacy or the actual extent of expression of the protein that's important? And as an example, for example, in the TCR field, for many years people have struggled to genetically modify T cells to express TCRs. Because you can get good transduction efficacy, but then you don't pick up your TCR on the cell surface, or you pick it at very low levels of expression. And it took at least 10 years or more before people started understanding this sort of weak TCR, strong TCR concept. And while they were baffled by why do I get so much vector into the cells are transduced, I can put a reported gene and all of the cells are positive, but there's not enough TCR. And so that would take things, it would slow things down quite considerably until you come up with some understanding as to, the gene that I'm expressing, a TCR, is actually a heterodimer of an alpha and a beta chain, or gamma and the delta. But then the alpha and the beta has the complex with CD3. There's 5 chains there. There's quite a lot of complex that needs to be put in place. And before you understand that, you think, well, what's potency? What functional characteristic am I looking at, or I should be looking at? Is it transduction efficacy? Is it expression? Or is it potency in terms of activation? Does it kill tumor cells? And that's why really we partnered with UCL, because we think this is the issue that they address, the expression of the TCR and understanding the TCR biology. And it did have to take a circle of about 20 years before you end up with starting to try and transduce cells with a TCR to starting to have an understanding as to how this whole thing works and eventually kills or doesn't kill tumor cells. And I don't think you would have been able to achieve that without having going in that 20-year circle and understood what's important and what's not important.
Yeah, so I completely agree with you, Stefan. I think then there's a lot of things we don't understand and we can't understand from in vitro potency assays, which really define how the product's going to function in the patient. But we need to try. We need to measure the things that we can, and actually, doing things like in vitro cell killing assays for cells kind of makes complete sense. In the end, what we don't know is whether if we always throw in for very high transduction efficiency, actually, well, that's a good thing. So we need to put specifications around what we think is reasonable. But we know if we get some healthy cells transduced and they meet their target antigen in the patient, those cells can actually proliferate hugely and expand on that. So just because you have a low transduction efficiency doesn't mean you won't have an ineffective product. So it's not as easy as it must be transduced, and that's the answer. But obviously you can measure it. You should try and measure what you can, but then there is still going to be variability in the patient.
Sure. Do we have any questions or comments or thoughts in the audience?
There we go. You ready?
Nice.
It'd be embarrassing if I wouldn't have caught that. I sort of had 2 related questions, so you can pick whichever one you want. When you look at either a technology that's coming down the pike, be it analytical or manufacturing technology, or something that you say, well, if I could do that, that would be great. So either one, is there something where you say, wow, if we could really do that, that would make a big difference, or here's a new technology coming down the pike that I really think is going to impact us in a positive way. Maybe you could opine on the future a bit.
Okay, it's like I'm volunteered to start. So, for me, I think, going back to my theme about the importance of characterization and analytics, I think improved inline analytics during the manufacturing of a cell-based product, I think, would make a huge impact. For example, when we're manufacturing for autologous therapies, we know that these patients, when they come in, depending on the pre-treatments they have, they've got different numbers of cells at starting point, and we know those cells actually will have different kinetics of growth. So actually being able to have inline analytics, so we really know what's going on in our culture systems on a kind of hourly basis, would actually enable us to really know how those cells are growing, enable us to introduce then adaptive controls to say, okay, this patient's cells are growing really well, we're going to be able to to shorten the manufacturing period. We've got the target number of cells. We've got good health. We don't have exhaustion happening. For another patient, actually, it's going to take longer. We need to feed more frequently because these cells need encouragement. So I think that kind of inline analytics can make a huge impact on the efficiency and the ability to always successfully manufacture for autologous-based therapies.
Yeah, I'll probably cop out of the question. I'm just a BD guy. But no, I think there's not really one big thing that I could point my finger and say, if we fix this, then everything is going to be fine. Because of the complexity of the therapeutics that we're developing, and certainly with T cells, you need to have a population of different types of cells there that each work with each other and help each other. And then end up having a proper immune response, which is then regulated properly. I think it's a matter of just tweaking the process. Starting with the parameters that one thinks are important, and then keep tweaking. I think we're more in the tweaking stage now, rather than the sort of the prototype stage. I like to think that we're slightly one step ahead of the prototype. We're sort of Ford Model T territory, where we sort of take very early prototypes and industrialize them in a way that you can make it roughly the same every time, and start to understand. You could look at the Ford Model T and the modern car. 4 wheels, steering wheel, and engine, usually at the same place. 4 seats.
Any color
Exactly. I don't know if they had another color available to them back then. But there's the modern car, which compared with an electric car, is still the same concept, but everything has been optimized from performance to fuel efficiency to safety to smart cars. So that's where I think we are now. Where instead of what came after Ford Model T is probably the next product that we will be selling. And as an industry, we're going to be making electric cars at some point, very, very far from now. But at least we understand every step of the way what we did, how it impacted the product, and then how it moves forward. Yeah. So it's tweaking, is optimizing. That's what I want.
I think it's really interesting. He was going to use a car as an analogy because I was going to use a car as an analogy. But I was going to say it's like we found a Ferrari with the set of keys, and we know it can drive us over there. But we have no idea how it works on the inside. We are a whole pile of reductionist trained scientists in an analytical world, or an analog world. Sorry, in an analog world in which we don't know enough about the systems biology. An old example is back in the day when I was early on the bone marrow transplant world. It was all about finding whatever that one stem cell is that could repopulate, hematopoiesis, and we found it. But it didn't work on its own. It worked in combination with a whole bunch of other stuff that had to be injected at the same time. We are still, I think, at that point where we're trying to industrialize the process and control the process as much as possible. But fundamentally, it's going into a biological system, and we don't know enough systems biology to make a lot of those decisions done well. So I think we need, in some ways, the systems biology world to kind of catch up to us, or we use it so we can make more comprehensive decisions, and we don't go down the rat hole of, oh my gosh, I got to look at glucose metabolism for this cell because that's the only thing that matters, because we don't know it matters, So I think that's where we're, if there's one thing we need in the next 5 years, it's this ability to do a systems approach towards both the patient, the product coming in, and the product going out.
Do we have any more questions as we're coming to the close of the workshop? Okay. All right. I want to thank you all for joining us today. Thank our panel for sharing and opining. Sarah, did you have a?
Yep. Okay.
Okay. All right. Thanks, everyone.